Welcome back to the deep dive. Today, we're really getting into the heart of chemical safety. Our mission, to understand how two key fields, toxicology and epidemiology, work hand in hand. They're basically our main defense against chemical risks we might not even see. We want to unpack how that partnership functions.
It's a really vital partnership, actually, because, well, you need both perspectives, don't you? You need the controlled lab results and the real-world observations from populations. Otherwise, you only get half the story on a chemicals risk. Maybe we should start by, defining what each field actually does.
Good idea. Let's nail those down.
Okay, so toxicology. That's the study of the bad effects chemicals can have on living things. It covers everything from symptoms to how the chemical works inside the body, its mechanisms, and even detection and treatment.
Right, so toxicology is really digging into the how. How exactly does this substance cause harm?
Precisely. And epidemiology. That takes a wider view. It's looking at the human population level.
The bigger canvas, as it were.
Exactly. Epidemiology studies the patterns of ill health, across large groups of people. Often this means using long-term studies, sometimes called longitudinal studies.
Like tracking people over time.
Yes, either looking back, those are case control studies, seeing who got sick and why, or tracking healthy people forward for years, sometimes decades, to see who gets sick. Those are prospective cohort studies.
Okay, so we have the lab science and the population science. Now, how do they connect for classifying chemical hazards? Our sources stress that toxicology is, well, the main driver initially. The classification into different hazard levels relies pretty heavily on toxicology data, right? The sort of hard science from labs.
That's generally true, yes. It's based on controlled experiments, often using things like animal testing. You might have heard of the fixed-dose procedure, or increasingly alternative methods like computer modeling.
And toxicology has specific tools for this.
Oh, absolutely. A key one is the dose-response curve. It's a fundamental concept. It graphically shows the link between how much of a chemical someone is exposed to the dose and the health effect of response.
And that curve gives us something specific.
It does. Crucially, it lets scientists figure out the no observed adverse effect level. We call it the NOAEL for short.
NOAEL. Okay, and that's important because...
Because it's the level of exposure where, based on the study, no negative health effects were seen.
Ah, so that's where the rubber meets the road for safety rules.
Exactly. Once you have a NOAEL, regulators can use that information to set protective limits, like occupational exposure limits or OELs, for workplaces. It gives a starting point.
A scientifically derived starting point for saying this level should be safe.
Right. But, and this is critical, that's not the whole picture.
Yeah.
This is where epidemiology becomes so important. It acts like a safety net. Toxicology classifies risks based on what we test or predict. Epidemiology helps us catch the things we didn't predict, the links that only show up in real human populations living real lives.
Okay, hold on. If toxicology gives us these predictive NOAELs from fast lab tests, why do we need these really long, sometimes decades-long epidemiology studies? Doesn't it seem a bit slow? Or like we're just checking if the lab tests failed?
It might seem slow, but it's not really about failure. It's about validation and, importantly, discovery in the real world. Think about classic examples like asbestos.
Right. Asbestos is always mentioned.
The alarm bells didn't first ring because of a lab test result. It was doctors, health professionals, noticing strange clusters of very rare lung cancers, like mesothelioma.
In specific groups of people.
Exactly. Often people who worked in shipyards or certain industries.
Yeah.
That pattern, that observation at the population level, that was the epidemiological trigger. It raised the question.
I see. So the real-world pattern comes first sometimes.
It can, yes. And then that finding prompts more focused toxicological investigation.
Yeah.
To confirm the link, to understand the biological mechanism of how asbestos caused that specific disease.
And once that causal link was confirmed by the science...
Then you get action. Stronger regulations, bans, ways to actually protect people based on solid evidence connecting exposure to the disease. Lead in petrol is another similar story.
So it really shows how they complement each other. Toxicology gives the primary data for classification.
And epidemiology makes sure we don't miss risks playing out in the complex human environment. It catches overlooked links.
Okay, so let's wrap this up for you, the listener. What's the key takeaway? You now have a kind of shortcut understanding of this dual system. Scientific testing through toxicology provides the main hazard classification data we use up front.
And real-world observation through epidemiology acts as that crucial check, ensuring we identify and address human health risks, even ones missed by initial lab tests.
And it's worth remembering that real-world observation part isn't always quick.
Not at all. Lab tests can be relatively fast, but some epidemiological studies, especially for diseases that take a long time to develop after exposure,
Hmm.
well, they take patience, immense patience.
We're talking decades sometimes, right? Like with asbestos and mesothelioma, it could be up to 40 years between exposure and disease.
That's right, 40 years.
Wow. That leaves you with a pretty sobering thought, doesn't it? It means we could be exposed to things today whose full impact on human health won't be truly understood until, well, maybe until people listening now are nearing retirement.
It really underscores the need for both the predictive power of toxicology and the long-term careful vigilance of epidemiology.
Speed and patience working together. Okay, that's our deep dive for today. Thanks for joining us.